Hello my dear students, myself Dr. Pitambar Humane, founder Botany for You and working as Professor and Head Department of Botany, Dharampeth MP Dev Memorial Science College, Nagpur, Maharashtra, India. I welcome you all in this online platform for the students of Life Sciences Botany for You. Friends, in this series of lectures in genetics and molecular biology, we have completed more than 20 lectures, and in today's lecture we will see the RNA, that is ribose nucleic acid, structure and types. Now if you see the RNA, that is the ribose nucleic acid, earlier we have seen the DNA, that is the deoxyribose nucleic acid. It means that there is only the difference of the sugar on the basis of this name, ribose sugar and deoxyribose sugar. In addition to that, the DNA is double stranded, but RNA is single stranded. So this is again one of the difference in between these two. The chemical composition of RNA is same to that of the DNA, that is it contains the phosphate in the form of the phosphoric acid, then sugar in the form of the ribose sugar, and the nitrogen bases just like to that of the DNA molecule, but only one difference, that in the DNA molecule there is a presence of ATGC and in RNA AUGC, means uracil is present in the RNA and thymine is present in the DNA molecule. So if you see this primary structure of the RNA, then in a primary structure it is just a single strand made up of the phosphate, sugar and nitrogen bases. As we have seen earlier in the DNA also, here the sugar, that is the ribose sugar, then phosphate and nitrogen base, again such type of the structure will be there, that is the chemical structure, and in this way here there will be formation of the single strand of the RNA molecule. Either the nitrogen bases will be single ring or the double ring. If it is the uracil, then cytosine, guanine and adenine. So such a type of the single strand structure is there in the primary structure. But even there is a formation of hydrogen bonds in between the complementary nitrogen bases present on the same DNA strand as it is a single strand. Rarely double stranded RNA is also present, but it is only in the viruses and not in the other organisms. So in such a cases there is again hydrogen bonding in between the complementary nitrogen bases, otherwise the single strand folded itself and forming the secondary structure because of the formation of the hydrogen bonds in between the complementary nitrogen bases. If we see this sugar, that is the ribose sugar and the deoxyribose sugar, only there is a difference of the oxygen atom at the second carbon atom, that this one is the first carbon atom, second carbon atom, third carbon atom, fourth carbon atom, and this is the fifth carbon atom which is present outside the ring. Now if you see this hydrogen, here there is a presence of such a type of the OH. Now if this OH is present, then it is a ribose sugar, and if this is oxygen is removed, see the name deoxyribose sugar itself, it means the correctly said that 2-deoxyribose sugar. To indicate the second carbon atom, then the D means the degeneration, oxy means the oxygen atom from the ribose sugar. So if here the OH is present, it means that it is a ribose sugar, and if this O is removed, that is the oxygen is removed, then it becomes now the deoxyribose sugar. So here at the second carbon atom, there is a presence of OH in the Rna molecule, that is the ribose sugar. And as I told you, the type of nitrogen bases which are present in the RNA, they are the adenine, uracil, guanine and the cytosine. If we see this nucleic acid, that is the RNA and its synthesis, usually in most of the organisms, it is synthesized with the help of sequence of nitrogen bases on the DNA molecule. And we know the DNA is present in the nucleus. In addition to that, it is found in the mitochondria and the chloroplast also. So this DNA molecule is used for the formation of the RNA, and that is known as the transcription, that we will see in upcoming classes. Such a type of the process in which the DNA template is used for the formation of the RNA, then it is known as the DNA dependent RNA synthesis. But if we are talking about the certain viruses like the tobacco mosaic virus, this is a RNA virus, means it contains only the RNA and not the DNA, which is the characteristic feature of the viruses. Now, if only the RNA is present, it means that here the RNA will be synthesized on the RNA, and such a type of the synthesis of the RNA as a RNA template, then it is known as the RNA dependent template. This figure indicates that the double stranded DNA molecule, out of that the one of the strand of the DNA is taken as a template for the synthesis of this RNA molecule, and in this way this is an example for the DNA dependent synthesis. So in the detail we will see it in the transcription. Now if we are talking about the types of RNA, very commonly we know there are the 3 different types, but if you see there are more than 10 types of the RNA which are found in the different organisms and are involved in the different functions in the prokaryotes and the eukaryotes also. Basically they are categorized here into the 2 types, that is the genetic RNA and second one is a non-genetic RNA. Earlier we have studied that the DNA as the genetic material as well as the RNA as the genetic material. So in most of the organisms the DNA is a genetic material, but if we see certain viruses, it contain either RNA or DNA. In such cases, those viruses which contain RNA, they are provided with the genes in that RNA molecule. It means that this RNA is also a genetic material for such type of the viruses, which is known as the genetic RNA. And this RNA, which is genetic RNA, is found in the form of either single stranded structure or even the double stranded structure, and on the basis of that again there are the different type of the viruses, that is the single stranded RNA viruses and the double stranded RNA viruses. While in other organisms except the viruses, there is presence of RNA and it is non-genetic RNA. Basically we are very much aware about these 3 different types and which are the part of the UG syllabus that we will see in the detail. That is the mRNA, that is the messenger RNA, second one tRNA and the rRNA. The messenger itself, the name is given on the basis of that it carry the message from the nucleus to the cytoplasm for the synthesis of the protein. Then transfer RNA is involved in transport of the amino acid for the protein synthesis. And the ribosomal RNA, the name indicates that they are present in the ribosomes. In addition to these three types, again there is a snRNA, that is the small nucleolar RNA which is present in the nucleolus. Then heterogeneous RNA, then small nuclear RNA, then antisense RNA, microRNA, small interfering RNA, then ribozyme, that is the catalytic RNA, and TM, that is the transfer messenger RNA. So these are involved in a different processes or metabolic processes in the different organisms. Basically, we are dealing with these three types of the RNA in this lecture. Now, first one, that is the mRNA. If you know the structure of the cell, then in a eukaryotes there is a presence of the nuclear membrane and the DNA molecule is present in the nucleus. So this is responsible to carry the genes. It means that the information which is present on the DNA in the form of the genes is utilized for the formation of the protein in the cytoplasm. And hence, if the process of the protein synthesis is carried out in the cytoplasm but the DNA is present in the nucleus, there is a necessity to transfer this information from the nucleus to the cytoplasm. And hence, first of all, this DNA is copied, but not in the form of DNA but in the form of RNA. And such a type of the process in which the RNA molecule is formed with the help of one of the strand of the DNA, and that is known as the transcription. I already told you that this topic of the transcription in detail we'll see in the topic of transcription. It means that when out of the two strands, one of the strand is used for the synthesis of mRNA in nucleus. So in this way, here after transcription we will have the mRNA in the nucleus. Now after formation of the mRNA in the nucleus, what will happen? Here we will have the complementary nitrogen base pairing of the mRNA to that of the DNA molecule. It means that here the same information is transferred but in a different way, as here are the nitrogen bases, that is the ATGC, and on the mRNA there is a AUGC. So there is conversion of the script in the form of mRNA by the process of the transcription. Now after synthesis in the eukaryotes there are the many changes, that is the capping is carried out at the 5' end with the guanosine and the poly A tail is added to the 3' end, and in this way even in eukaryotes there are the exons and the introns. So or the coding and the non-coding regions. So the coding regions are kept and the non-codings are removed, and in this way final mRNA copy is generated which is ready for the protein synthesis. So in this way such a type of the mRNA is formed with the help of single strand of the DNA, that is known as the template. Now this sequence of the nitrogen bases which is present on the mRNA will be utilized for the synthesis of the protein, and the synthesis of the protein will be completely dependent upon the sequence of nitrogen bases present on the mRNA. And if you see the sequence of this nitrogen bases of the mRNA is the reflection of the sequence of nitrogen bases on the DNA. It means that whatever the genes which are present in the nucleus, the same type of the protein will be formed in the cytoplasm. And for that there is necessity to transport the message from the nucleus to the cytoplasm by the RNA in the form of mRNA, and hence it is known as the mRNA. In some cases only the one gene is transcribed, that is the one cistron is transcribed. In such cases mRNAs are known as monocistronic. But in some other cases there is need of the multiple proteins to be synthesized for a particular processes, and hence the many cistrons are transcribed at the same time for the single mRNA. And such type of the mRNA which are responsible for the synthesis of the multiple genes or copying the multiple genes, and that are known as the polycistronic mRNAs. The second type, that is tRNA. T indicate the transfer, or the sRNA means the soluble RNA. It is unlike to that of the mRNA. It is folded structure and having also a complementary nitrogen base pairing. This is actually the structure of chloroleaf model. So this indicates 3 leaves, and this is the stalk or the petiole. So in this way it shows the structure similar to that of the chloroleaf, and hence it is known as the chloroleaf model. As the name indicate the transfer, it means that it is involved in the transfer of amino acids. Now why there is necessity to transfer this amino acid? If you see the proteins in the living organisms, all the proteins are made up of 20 different amino acids, and that particular amino acid is formed on the basis of the sequence of particular amino acids. It means that this sequence of amino acid is very much important for the formation of the particular protein. And these amino acids, when are added during the protein synthesis, they are transferred by the tRNA molecules. It means that these 20 different amino acids will require minimum 20 tRNA molecules. But if you see in the living organism, there are up to the 60 different types of the tRNA which are involved during the protein synthesis for the transport of the amino acids. Now, even because of a very small size among the RNAs, the tRNA is also known as the soluble RNA. This chloroleaf model is distinguished into the T-size loop, then DHE loop, and the anticodon loop and the variable loop. The name indicates the length in the different organism or in a different tRNAs is different, and hence it is known as the variable loop. The anticodon loop, it means that here the sequence of 3 nitrogen bases which are present on this loop, they are used as a anticodon. If you see the codon, that is the sequence of 3 successive nitrogen bases present on mRNA, and that is known as the codon. And the complementary nitrogen base pairing of the codon present on the anticodon loop, and that is known as the anticodon. So such type of the 3 nitrogen bases which are present on the anticodon loop, they are considered as a anticodon, and hence on the basis of this sequence of nitrogen bases in the form of anticodon on the tRNA, here it can recognize the sequence of nitrogen bases present on the mRNA, that again we will see in the upcoming lecture of the translation or the protein synthesis. It means that there is necessity to have the complementary base pairing in between the codons and anticodons. The codons on mRNA and the anticodons on the tRNA. And on the basis of this particular type of the sequence of anticodon, again here there is a acceptor stem where the amino acid get attached. So that is known as the amino acylation. So this amino acid, that is the particular 20 amino acids are there in the living organism, so that there will be the need of the 20 different tRNA molecules to carry these amino acids. It means that here the tRNA molecule is also playing an important role in protein synthesis, as the particular amino acids are carried out by the particular tRNA molecules. And this is completely dependent upon the sequence of nitrogen bases present on the mRNA, and that is read by the anticodon of the tRNA molecule. The third type, that is the rRNA, the name itself indicate the ribosomal RNA, as they are present in the ribosomes. During protein synthesis, the ribosomes are attaches itself to the mRNA, and hence these RNA molecules which are present in the ribosome, they are responsible for this attachment as well as to recognize the codon anticodon sequence. So this is mRNA and smaller subunit of the ribosome and the larger subunit of the ribosome. So in the smaller subunit as well as the larger subunit, there are the presence of the different RNA molecules which are known as the rRNAs. Usually these rRNA are present single stranded, but they are folded upon itself, and because of that there is a formation of the hydrogen bonding in between the complementary nitrogen bases. If we see the prokaryotes and the eukaryotes, then in eukaryotes there are the presence of 80S type of the ribosomes, and in prokaryotes there are the 70S, S for the sedimentation coefficient. These 80S ribosomes are made up of two subunits, that is the 60S larger subunit and 40S smaller subunit. And in a larger subunit there are presence of three different type of the rRNA, that is the 28S, 5.S and the 5S. And in the smaller subunit in the eukaryote there is a presence of rRNA, and that is the 18S. Likewise, in prokaryotes there is presence of the 70S type of the ribosomes which are made up of two subunits, that is the larger 50S and the smaller 30. And in a 50S subunit there is presence of two types of the rRNA, that is the 23S and the 5S, while in the smaller subunit, that is the 30S, there are the presence of 16S rRNA. These rRNAs are usually helping the mRNA, ribosome and tRNA to bind together during the protein synthesis. So friends, I am very much hopeful and confident that this lecture will be useful to you in preparation of your examination. In next lecture, we will see the genetic code. If you like this lecture and if it is useful to you, then kindly like, share and subscribe our YouTube channel Botany for You. And also press bell icon for the alerts of every lecture to be uploaded on different life sciences titles. And don't forget to join the Telegram group with the link given in the description, or even you can also visit to our website www.botanyforyou.com. Thanks a lot once again for watching this lecture completely.